Substances move across the plasma membrane in and out of the cell constantly. Sometimes they do so passively, meaning the cell does not have to expend any energy or ATP to drive the process, while active transport requires the cell to use energy or ATP to move substances across either in or out of the cell. This video will go through the different modes of passive transport and the different modes of active transport. Simple diffusion is a passive process, which means no energy is required from the cell to drive this process. Simple diffusion simply means that particles will move from a high to a low concentration, which is referred to as down the concentration gradient, as long as the plasma membrane allows passage. Here are some particles. They're in high concentration outside the cell and low concentration inside the cell. So over time, these particles will diffuse into the cell from high to low concentration or down the concentration gradient. Particles are still moving out of the cell by the time we get to this time period, but particles moving in will equal particles moving out. So there's no net movement of particles at this point. To recap, simple diffusion is movement of particles from high to low concentration, down the concentration gradient. Granted, the plasma membrane will allow passage through. Facilitated diffusion is also a passive process. No energy or ATP is required by the cell. It's still diffusion, therefore particles move from high to low concentration. But with facilitated diffusion, particles cannot get through the plasma membrane. They require some type of protein channel to allow passage. There's different types of passages that can occur with facilitated diffusion. A channel protein, like this one, will allow particles to move from high to low concentration. A carrier protein is almost the same. The only difference is that it will change shape when a particle enters the channel to facilitate the movement, in this case, into the cell. Specific channel proteins that allow water to pass in and out of the cell are called aquaporins. These are all types of facilitated diffusion and require no energy and are therefore a passive process. Osmosis is another passive process that does not require energy or ATP to be expended by the cell. Osmosis is diffusion, but diffusion of something very specific, water molecules. Since it's diffusion, water moves from high to low water concentration. But that's hard to picture because water is typically the background when we look at an image, not the particles we see in the water. So it would be easier to think of it this way. Water follows solute, for example, salt. If we look at this image, we see that on this side, there's very little solute, where on this side, there are a lot more solute particles in the solution. So if we use our little saying here that water follows solute or salt, water will go from where there's less solute to where there's more solute. So water will move from the left side of this beaker to the right side. So in this image here, we see that water has moved to the right because it's following the higher solute or salt concentration. This membrane here is semi-permeable, which means it allows passage of some particles but not others. In this example, water is allowed to pass through, but the solute, whatever it is, it could be salt, is not allowed to pass through. Let's see how osmosis applies to our blood. Let's look at the center image first. It's an isotonic solution, which is the ideal situation. We have our red blood cells, which are suspended in our blood liquid, plasma. The concentration of solute or salt inside the cell equals the concentration of salt outside the cell. Iso means same. So isotonic means same concentration of solute inside and outside of the cell, and you can see that here. In red, we have the solute, which happens to be salt, sodium chloride, and the concentration is equal inside and outside the cell, which means water will diffuse out through osmosis, water will diffuse in through osmosis at equal amounts. Therefore, there's no net movement of water, and the cells stay the same shape. They keep their structure, which is important to perform their function, that being to carry oxygen to all of our cells. Let's look at a hypertonic solution. Hyper means above or excessive, and if we look at this image, we see these red dots, these salt particles, in high concentration outside the cell, but in low concentration inside the cell. Recall from last slide that water follows salt. So where is the higher salt concentration? It's on the outside. Therefore, water will diffuse from inside the cell to outside the cell. Of course, some water will diffuse in, but not as much as the water diffusing out. So the net movement of water in a hypertonic solution is from inside the cell to outside the cell. And we can see what happens to the cell in a hypertonic solution. These red blood cells start to shrink, and we call that very specifically crenation. We can also say the cell crenates. A hypotonic solution requires more salt or solute on the inside of the cell versus the outside of the cell. Since water follows salt, water is going to move into the cell. Of course, some water will leave the cell, but nowhere near as much as the amount of water molecules moving into the cell. So when we're talking about osmosis in a hypotonic solution, hypo means the solution is below or deficient in solute. We see that water moving into the cells will cause the cells to swell. If they swell enough, they could burst. We call this hemolysis. Hemo refers to blood, and lysis refers to rupturing. Active transport has to do with particles moving across the membrane, but requiring energy to do so. A good example of this is the sodium-potassium pump, and we're talking about sodium ions, which we see in orange, sodium with a positive charge, and potassium ions, which we see in yellow. That's potassium with a positive charge. We see that sodium in orange is in high concentration outside of the cell and in low concentration inside of the cell. So diffusion says it will want to diffuse in from high to low concentration. However, in active transport, the cell is going to expend energy to pump sodiums to the outside of the cell. Potassiums are in high concentration inside the cell and low concentration outside the cell, so they'll want to diffuse out, but the cell will expend ATP in order to pump potassium ions into the cell. This is the sodium-potassium pump. An important note here, which will be important later in the course, is that 3 sodiums get pumped out for every 2 potassium ions that get pumped in. Since they both have positive charges, more positive charges are leaving than entering the cell. Therefore, the outside of the cell becomes more positive compared to the inside of the cell, which is more negatively charged. Endocytosis is another active process which requires energy. there's three types. Endo means within, cyto means cell. Therefore, endocytosis simply means into the cell. And that's what's happening. One type is phagocytosis. Phago means eating, and that's when the plasma membrane engulfs a large particle and takes it in. The little bubble is called a vacuole. It's like the cell is eating, which is why it's called phagocytosis, eating or swallowing. Pinocytosis is a type of endocytosis as well, where the plasma membrane will pinch in to take in small little droplets of water into a little vesicle. That's analogous to cell drinking. Pino means drinking. So pinocytosis is when the cell takes in small droplets of water. Sometimes the cell is very specific in what it wants to take in, so something has to bind to a receptor, but only what that receptor is looking for. When that happens, the plasma membrane pinches in, and we have our very specific molecule being ingested into the cell. This is called endocytosis, but receptor-mediated endocytosis, because this plasma membrane, these proteins on it, are very picky on what it allows in. So receptor-mediated endocytosis, pinocytosis, and phagocytosis are all types of endocytosis, which is taking in material into the cell. All these processes require energy, so all these processes are active. Exo means out. Therefore, exocytosis is the process of moving material out of the cell. It's just the opposite of endocytosis. We have particles in a vesicle, which make their way towards the plasma membrane, and that vesicle will release the product out of the cell. Exocytosis is an active process as well, and therefore requires energy or ATP expenditure from the cell.